Okay, so here we are almost done with chapter 21 now. We've talked a lot about nucleic acids and protein synthesis. We've talked about what happens when things go right. Our last lecture was what happens when things go wrong. And this has all been pretty much up to nature aside from some of those mutagens that are human-made from our previous lecture. So now we're ready to move out of what nature has done and into where humans have started to tinker with things. Okay, so the particular focus of this lecture is recombinant DNA, and this is where humans get involved and decide to do things like splicing DNA of one species into another, and sometimes the results are good, sometimes the results are not so good, and we'll talk about those successes and failures throughout this lecture. Okay, so in recombinant DNA, a DNA fragment from one organism is combined with DNA in another. We have restriction enzymes that are used to cleave a gene from a foreign DNA and open these DNA plasmids in something like E. coli. And the DNA fragments are mixed with the plasmids in E. coli, and the ends are joined by ligase. We talked about DNA ligases before, so this is just to zip it up. And then the new gene in the altered DNA produces protein. So typically we can have something that would not ordinarily be produced by E. coli, for instance, silk protein. We might want to try to splice in the genes from the silkworm to try to cause the E. coli to produce silk protein. That would be great, wouldn't it? You could have silk prices go down drastically as the silk is produced much cheaper and more easily by the E. coli. So here we see a cartoon of that where we have the restriction enzyme that cuts the plasmid DNA at a certain place, and now it's got sticky ends where we've spliced it there, and we can take those DNA plasmids and have our restriction enzyme cut the plasmids. We get the bacteria with the recombinant DNA plasmid. We can rejoin it, and we can create these cloned cells that will produce new enzymes, such as new proteins rather, such as enzymes or hormones. The example I gave, they haven't done it in bacteria to my knowledge, but they have used some transgenic DNA technology to get goats to produce silk in their milk. So lots of interesting things going on. Sometimes it doesn't work out so well. If you remember our cloned sheep Dolly, Dolly didn't last too long, and for whatever reason the clones don't tend to last as long as the regular non-cloned organisms when we get to larger things like sheep. But for the cloned cells here, when we're dealing with bacteria, things seem to go very well indeed. And so some of the successes here are products of recombinant DNA for therapeutic use. So human insulin, for instance, is used to treat diabetes. Erythropoietin treats anemia, stimulates production of erythrocytes. Human growth hormone stimulates growth. Interferon there for treating cancer and viral disease. And just on down the line to look at these different applications here of recombinant DNA technology. So lots of good things. It's not a whole lot of Frankenstein type stuff. It's actually very good and very safe science that results in true and measurable benefits to human life. We can also use DNA technology to do something called DNA fingerprinting. So for the southern transfer type of DNA fingerprinting, you have restriction enzymes that cut a DNA sample into smaller fragments, restriction fragment length polymorphisms, as they're known, RFLPs. The fragments are then sorted by size. You can have a radioisotope that adheres to certain base sequences in the fragments to produce a pattern on X-ray film, which is called the fingerprint itself. And that fingerprint is unique to each individual DNA. So this is one way that you can determine the identity of, say, a criminal in a forensic setting is through DNA fingerprinting. Of course, this would rely on you having lots of DNA of that suspect at the scene. In keeping with our forensic theme, let's look at polymerase chain reaction, PCR. That can produce multiple copies of DNA in a short time. So if you didn't have enough DNA of a suspect, for instance, at a crime scene to do the traditional DNA fingerprinting approach, what you could do is take the little bit of DNA you might be able to recover and then use polymerase chain reaction to multiply that DNA. So then you can separate the sample DNA strands by heating. So this is how you're able to cause these DNA samples that are actually at the scene to magnify in amount by doing what biology does, doing what nature does. You heat them up a little to get them to overcome those hydrogen bonds that are keeping them together, mix the separated strands with enzymes and nucleotides so that they have the basic stuff they need to make new complementary strands, and then you can repeat that again and again, right? Every time, if you start with just a single double helix, you separate it, now you can make two samples from that one DNA sample, and then you can take those two double helix, unwind them, now you can make four, and so on and so forth, and you can just magnify things exponentially in a very short time. And so here we see a cartoon of that process. So if we have just the one pair, the one DNA double helix, we can go ahead and heat to separate the strands. We do some primers to get the complements to form. And with that first cycle, we took that one double helix and now we made two. And then if we go through the process in a second cycle, we can have four, and so on and so forth, all from that one original. We can very, very rapidly, within a couple of hours, make many, many millions of copies. So DNA fingerprinting is also known as DNA profiling, and small samples of DNA obtained from body fluid like blood or skin cells or other body fluids like saliva or semen are good sources of the DNA. The DNA amount is typically small in those samples, so you can amplify them using the PCR polymerase chain reaction method we just described. The restriction enzymes can then cut that sample into the smaller fragments, which we talked about earlier when we mentioned the Southern transfer DNA fingerprinting. So if you had enough to begin with, you could go right to it, but typically you're using PCR to magnify the amounts, and then you can go ahead and restriction enzyme cut the DNA into smaller samples, place that on a gel for separation. The DNA fragments get separated by size using electrophoresis of the gel, and then we've talked about this forensic application to connect a suspect with a crime. And so here we see we might have multiple suspects, and we see whose matches the particular profile at the scene, and that would give us an indication of which suspects we might rule out and which suspects we might want to go back and visit for some more information. So in addition to that narrow application in forensic science, we can also think about DNA technology in terms of the Human Genome Project, which went on for 13 years. So from 1990 to 2003, we were involved in the project. I wasn't personally involved, but scientists were involved. And the goals were to identify the approximately 25,000 genes in human DNA, determine the base pair sequences of human DNA, and store this information in databases accessible on the internet. So some important summary information so far, and again, just because the project ended doesn't mean we know a whole lot about what those genes are and what they necessarily do, but the scientists determined that most of the human genome is not functional, and that sounds counterintuitive, but that's actually a good thing, because every time your cells divide, the ends, the telomeres, have a little bit cut from them. So you need a lot of nonsense, a lot of non-functional genomes, so that you're not losing critical information there as you're losing the ends of those chromosomes with each cell division. The coding portion of the genes is about 1% of the total genome. That sounds ridiculously small, but again, nature has evolved that way for a reason. It probably is a very safe thing to preserve those actual functional genes across about 1% of that overall genome. And finally, the results of the project will aid in the identification of defective genes that lead to genetic disease. We already have a number of genes that we associate with certain types of cancers. If you think back to Angelina Jolie, she made the very bold choice to have her breast removed as a result of having a test that showed she had one of the genes associated with breast cancer. So she decided not to wait around and see if it would indeed lead to breast cancer. She used that information to be proactive and act on the information, just knowing that she had the gene that is associated with that sometimes deadly and always very difficult disease. So there you have it, our lecture on recombinant DNA. We don't have any learning checks for this chapter, so hopefully you've been paying close attention and you're ready to join me one last time for our final lecture in our final chapter. See you then.